Welcome to the Masterclass. For over a century, modern medicine has operated on a simple, reactive premise: wait for a disease to appear, and then finally attempt to treat it. By focusing entirely on treating the late-stage symptoms of individual conditions like diabetes or neurodegeneration, we have historically bypassed the biological aging process that makes those diseases possible in the first place. To understand how to intervene, we have to separate chronological age, the strict number of years on a calendar, from biological age, which measures the actual cellular wear and tear occurring deep within the body's tissues. To actively intercept this cellular decline, clinicians need a way to continuously track it. Historically, securing a precise readout of our internal biological clock required invasive, painful tissue biopsies. The answer to this diagnostic roadblock happens to be our largest and most accessible organ, the human skin. The skin acts as a highly dynamic biological sentinel. Because it is deeply intertwined with our immune, metabolic, and neuroendocrine systems, it gives us a direct, measurable window into our overall systemic health. Every day, our skin serves as our first line of defense against ultraviolet radiation, pollution, and climate variation. We call this total accumulated environmental stress the exposome. This continuous bombardment forces the skin to adapt, causing chemical modifications to our DNA over time. These epigenetic alterations act like a dimmer switch, changing how specific genes behave, turning healthy functions down or off entirely, without actually altering the underlying genetic code. To track these changes, researchers developed the proteomic clock. Because proteins operate as the functional machinery of the cell, we can sample the proteins left behind in the stratum corneum, the outermost layer of our skin, and calculate a precise, quantitative biological age metric. Because the specific skin layer naturally sheds on its own, clinicians can collect these crucial protein samples non-invasively, completely bypassing the need for surgical tissue extraction. By analyzing the subtle shifts in our skin's protein structures, we now possess an active, real-time diagnostic clock capable of tracking exactly how fast our internal systems are aging. When we examine the deeper cellular layers of aging skin, we find the distinct accumulation of zombie cells. This biological state is known as cellular senescence. It is a defense mechanism where heavily damaged cells stop dividing to prevent mutating or turning cancerous. But instead of dying and clearing out, they refuse to self-destruct and remain metabolically active. These senescent cells secrete a toxic cocktail of inflammatory molecules, a distress signal called the SASP, directly into the surrounding healthy tissue. This continuous cellular crosstalk triggers a persistent immune activation known as inflammaging. It is a chronic, low-grade fire that spreads from the localized skin layer straight into our bloodstream. Locally, this constant inflammation degrades the skin's structural integrity, accelerating the formation of wrinkles and the loss of elasticity. Systemically, it drives the steady, functional decline of our internal organs. Surface-level skin aging acts as our earliest visible warning sign. It reveals a deeply destructive systemic inflammatory crisis happening entirely out of sight. To combat this decline, we have to look at the skin's core energy factories, the mitochondria. As we age, the efficiency of these microscopic power plants plummets, stripping cells of the energy they need to repair themselves. This diagram illustrates what happens when those cellular energy levels drop. Without sufficient power, a critical quality control system called proteostasis collapses, leading to a dangerous buildup of misfolded proteins and cellular stress, compared to the functional proteostasis seen on the right. To intervene, researchers are utilizing targeted biological compounds, or bioactives, designed to actively reprogram our failing cellular machinery. Specific molecular interventions, like the postbiotic urolithin A and the polyphenol resveratrol, act as biological switches. They induce a recycling process that clears out dead, dysfunctional mitochondria and reactivates the energy pathways necessary for tissue repair. Scientists are also using advanced metabolomic profiling to decode ancient botanical sources. By analyzing the complex biochemical profiles of specific plants, researchers extract potent tissue regenerative properties that enhance mitochondrial function. Rather than passively observing the decay of our tissues, we possess the molecular tools to restore resilience and reverse cellular decline directly at the energy source. The underlying truth of longevity science is that the skin operates as a two-way street. It is directly tethered to our internal immune and metabolic networks. Because of this intimate connection, initiating a targeted cellular repair on the skin's surface cascades downwards, helping to suppress the systemic fire of inflammaging throughout the entire body. This exposes the precise limitations of traditional cosmetic treatments. Simply masking symptoms with topical creams does nothing to address the biological wear and tear driving the decline. These clinical insights define geromedicine, a discipline focused on treating the fundamental biology of aging with clinical precision and measurable data. Integrating multi-omic diagnostics and targeted bioactives allows us to fortify cellular resilience from the inside out. Protecting and decoding this biological shield provides the specific data required to extend human health span.